Hello, my name is Crystal Hammack, and the purpose of my presentation is to talk about cancer cells, what they are, how they're treated, some different treatments that they have looked into, some research, did it work, did it not work, and I actually talk about this topic from a little bit of experience also. So the objectives are going to be to explain the properties of cancerous cells, identify the genes involved in the cancerous cells, list current treatment options using molecular diagnostics and targeting of specific cancer cells. Understanding the properties of cancerous cells enables early detection and diagnostics. So what I would say from a personal aspect is go to your doctor regularly, have the checkups that you're supposed to have. If you think that there is anything wrong, get it checked out, because it's better to go and it not be something than to wait and it be so advanced that you're past where you should be. Guides development of targeted therapies, provides insight into potential metastasis. Identifying genes involved in cancerous cells. Well, here I picked the BRCA1 and the BRCA2, and these are genes that they look for when they're talking about breast cancer. So if you're going in and you're having any kind of testing for breast cancer, these are the genes that they look at, because I'm sure that you all know that there's more than just breast cancer out there, and well, there's a lot more than breast cancer out there. And it was hard to kind of just pinpoint one. So I'm kind of talking about more than one, but I didn't get to talk about all of them because this is supposed to be about 15 minutes. Identifying genes involved in cancer cells helps in predicting susceptibility to certain cancers, guides research for targeted drug treatment. And then I'm going to list some current treatment options using molecular diagnostics and targeting cancer cells. And the reason for doing this is because it allows for more precise and effective treatment. It minimizes side effects on healthy cells. It improves overall patient outcomes. It informs decisions on combination therapies for enhanced efficiency, and it promotes advancement in the field of oncology, because that's about what we do with science. We research, we see what works, we see what don't, and we go from there. Cancer is irregular cell growth. It can be invasive. It can spread to different parts of your body if untreated. Cells are required to continuously renew themselves in order for us to grow, in order to replace cells that have died, in order to replace cells that are not good. It's just the function of our life. And a cell can mutate in mitosis. And when it does, this can contribute to cancer, and it can form cancer as a tumor. So studying cancer and everything that is involved is very important. Doing research to advance what we know about cancer, how we get it, the best way to treat it without getting sicker than what we are, is very important. I picked this image over here on the side. It says cancer by the numbers, and it says 7.5% of average people that get cancer, it's inherited. 39% of Americans who will have cancer at some point. 15 million people will survive. So the number has grown over the years of who will survive when they hear the word cancer. So I could tell you from personal experience, hearing it it doesn't make it any less scary thinking how far we have come, but it did make it a lot more bearable knowing that I had options that wasn't just, let's go get chemotherapy, radiation, you're going to lose your hair and you're going to be sick. Because just like I am now, then I was in college. There's over 100 types of cancer that can affect humans. Leukemia and organ cancers are the most common. There is a high demand for chemotherapy and anti-cancer drugs. But some of you may know that those types of treatments can make you, well, for lack of a better word, sicker than what you were before you were actually sick. And I have known from doing the research from when I was sick that some people actually die from being treated with chemotherapy. They just could not come back from all the damage that it did to the rest of their body and just what it did to their immune system. So I think it is a good thing that we have the kind of research that we have, and people are still making advancements as of today. Right here we see what normal cells are supposed to look like, and these are different parts of your body. And then this is what cancerous cells look like. So they're misshapen, the nucleus is bigger, and I'm glad that somebody is able to look at a cell now and know that, hey, that cell does not look like it's supposed to. Let's see what's wrong with it. And then they did tests and they found out, hey, if a cell looks like this, then it's a cancerous cell. So as I stated a moment ago, there are over 100 types of cancer that impact humans. Leukemia is one of them, and this is a category of blood cancers. And then the cancers that affect organs would be your breast cancer, prostate cancer, lung cancer, liver cancer, cervical cancer, and they're prevalent. Consequently, there's a continual high demand for chemotherapy and anti-cancer drug delivery for these types of cancers. But there are advancements. There's nanotechnology-based anti-cancer drugs that deliver systems that specifically target malignant cells. This approach aims to eliminate or diminish proliferation of these cells while sparing healthy cells from harm. So with the chemotherapy, where it affects even your healthy cells, this technology is supposed to target the specific cancer cells only so that your body is able to heal. In my research of cancer, I found that Egypt does a lot of research with cancer because they experience high cases of liver and breast cancers in their area. They are committed to do research to find out how to better help the patients in their area. Their commitment is evident through integrated quality programs in patient care, research, education, and prevention. It's amplified by the establishment of the National Cancer Institute in the 1950s. As Egypt's largest comprehensive cancer center, the National Cancer Institute champions a multidisciplinary approach to cancer management. They emphasize collaboration among treatment and modalities. They address the disease management challenges by involving isolated genes associated with specific cancers, constructing gene regulatory networks, and identifying biomarkers through the changes in the GRNs. Modeling the GRNs is a complex multidisciplinary task that has gained significant attention in the recent years. As I said before, chemotherapy causes immune and organ damage. So a lot of people are trying to go away from that because of what it does. There are so many different cancer treatments out there, from chemotherapy, radiation therapy, targeted therapy, surgeries, immunotherapy, hormone therapy, stem cell transplant, and then active surveillance. That's just naming some of them, because I have heard of other ones that are basically just trials, and I don't really know if they work or not, because I just heard of what they were doing when I was going through what I went through, and I really didn't see a lot of those while I was doing the research, so I didn't really put them in here. But I know that they talked about some nanotech technology, and it offers a solution by enabling direct selective access to cancer cells. They have programmable nanoparticles that recognize and deliver drugs selectively to cancer cells, and this avoids interaction with healthy cells for more precise treatment. When I was doing my research, I got introduced to biomarkers. I had never heard of biomarkers before, and I thought that this was a really great thing to find. So I'm going to talk about that for the next few slides. Medical biomarkers are widely used, yet few were said to provide valuable for cancer diagnoses and monitoring. Thank goodness for modern medicine and them not giving up, because they said that they had no major breakthroughs in 25 years, but they did not stop doing the research. They spent tons of money on it, and they just were not finding what they wanted to find, but they didn't stop looking. Recent advancements in nanotechnology-based formulations have enhanced diagnostics and therapies for diverse diseases, notably addressing changes in cancer treatment. Existing chemotherapy, as I said before, was limited. It had lack of specificity for targeting just the cancer cells, so it attacked all the cells. Side effects were maximum in most cases, and it was actually growing to multi-drug resistance. Nanoparticle-based drug delivery overcomes these hurdles by improving drug stability, bioavailability, tissue penetration, and targeted delivery, enhancing efficiency against tumor cells. The ease of surface functionality and encapsulation properties facilitates the delivery of various antigens and tumor cell lysates in the form of nanovaccines, promoting and improving immune response. So they were getting somewhere because they did not give up. Additionally, these nanoparticles, with their smaller size and unique optical, physical, and mechanical properties, have evolved as highly sensitive and specific biosensors for detecting various markers, including nucleic acids, proteins, antigens, and small metabolites. So what changed in the biomarkers? They continued to research. They tried it on different cancers to see which ones it worked with, and found that it worked best with any kind of cancer that formed a tumor. So in between 2010 and 2023, biomarkers went from being a thought for promise to actually showing promise. Despite evolving regulatory frameworks over decades, the promise of biomarkers has shown minimal improvement in human health. Cancer's complexity makes unraveling the integrative and dynamic nature of the system through biomarkers challenging. But the past 2 decades, they witnessed advancements like multi-omics profiling, liquid biopsy, single-cell analysis, and artificial intelligence, promising a transformation in biomarker discovery. Combining the omics modalities aim to prove a comprehensive view for therapy selection and patient monitoring in precise medicine, especially in oncology. So to advance precision medicine, a shift from reductionist thinking to recognizing diseases as complex adaptive systems is crucial. Redefining biomarkers as representations of a biological system encompass traditional and emerging markers and complex algorithms. So I wanted to look at a specific example of how biomarkers have helped. And in China, they have hepatocellular carcinoma as one of the cancers that they see a lot of in their area. And it goes along with chronic hepatitis B and liver cirrhosis. There are serum proteomes that were analyzed from healthy controls, constructing the first cancer's trajectory for liver diseases. Their findings highlighted biological processes that were related to cancer hallmarks and potential therapeutic targets by changing the signaling pathway. Biomarker panels for the HCC, which was the hepatocellular carcinoma, was detected in the CHB, which is the chronic hepatitis, and the liver cirrhosis, as high-risk populations were developed using machines learning in 2 cohorts. The protein signatures significantly improved the HCC detection compared to using the traditional biomarker. Further validation with parallel reaction monitoring mass spectrometry in an additional cohort confirmed selected biomarkers. So this study provided insight into cancer biology processes in liver disease, offering candidate protein targets for early detection and intervention. So I thought to myself, when I thought about doing this educational presentation, not only what can I tell other people, but what can I also learn from it? Because that's one thing that I really enjoy doing. I like to learn. And I really was just going to look at what was going on in America, and I came across what they were doing in Egypt, and then I came across what they were doing in China. So I called this slide Research on the Move Again because I moved to a different country to see what they were doing in other places. And I came across Japan. They hosted the 15th International Gastric Cancer Congress by the International Gastric Cancer Association. Over 1,300 experts from 44 countries addressed key issues in stomach cancer, with over 800 abstracts submitted for presentation. So all of these oncologists got together with other scientists and other doctors and said, what can we do, not just where we live, but across the world, and share what we're doing so that we can get rid of cancer and make people healthier overall when it comes to this. So despite what we thought was a slowdown in scientific research, especially when the pandemic came around, this omics-based research in this gastric cancer aligns with the global advances in the human genome research. Notably, trends in gastric cancer genome research encompass the shift from a single reference genome to the reference pentagenome transitioning from a single population to cross-race studies. The focus has expanded from cancer cell-centered to a combined approach with microenvironment. So now, where in the beginning, when we started learning about cancers and you can get cancers from the environment, they went farther into the environment, in my opinion, because we went to the microenvironment. And before, we just looked at the large scope of things like cigarettes causing cancer and certain plastics burning causing cancer. So we went from next generation sequencing with short reads to third generation sequencing with long reads. And now we're going to move on to look at a variety of cancers that have been looked at. Moving on from biomarkers, I found something called immune checkpoint blockade, and it emerges as a promising strategy in the treatment of advanced cancers, offering substantial survival benefits across various cancer types. However, only a small proportion of cancer patients experienced positive responses and benefits from the immune checkpoint blockade therapy. The effectiveness of it was hampered by numerous factors, with intricate tumor microenvironment playing a crucial role. A comprehensive understanding of intricate tumor microenvironment composition is vital for unraveling the mechanisms behind varied responses in ICB among patients. Single-cell RNA sequencing, a novel technique, unveils gene expression profiles at the individual cell level, enabling a high-resolution exploration of the intricate tumor microenvironment heterogeneity and identification of key cell subsets, influencing the immune checkpoint blockage response. They say that this review underscores the significance of the ICB response because they're still doing research on it, and they're discovering that with the biomarkers predicting the ICB response and potential therapeutic targets for immunotherapy, that these findings are going to offer insights into future directions for implementing cancer immunotherapy in clinical settings, fostering advancements in precision medicine. So just like before, they're not seeing the success that they wanted to see. There are some setbacks, but they believe that they're going to have a breakthrough. And this is the most current data that I've seen in cancer research. With that being said, I put a few links in here for some additional articles if you wanted to see some things that are happening in cancer. And then I have my references right here at the end. And I will tell you part of this educational experience, other than just the cancer cells, is learning how to reference things properly, which I'm not that great at, especially when it came to the pictures that were on the slides. But if you have any questions, I think that you're going to be able to put them in comments, and I will try to answer those as they come in. And I hope that you all learned something from my presentation. I know that I sure did, and I look forward to reading y'all's.